we can understand the chemiosmotic hypothesis of ATP formation with this diagram. This is the diagram of thylakoid membrane. This is thylakoid lumen, and this one is the stroma of the chloroplast. Okay. Now, as we know that the thylakoid membrane having photosystems. Photosystem 2 and this one is a photosystem 1. Near the photosystem 2, towards the lumen side, oxygen evolving complex is present. Whereas near the photosystem 1, towards the stroma side, NADP reductase enzyme is present. Ferredoxin FMN. Here you can see this will take proton from stroma and reduces NADP into NADPH2. That means here it will utilizes proton from the stroma. Now, when the light rays falls on PS2, this energy received by this reaction center, the electron get excited and it will transfer to the electron carrier into the membrane. So this electron carrier, that is plastoquinone, is also work as a proton pumping or hydrogen carrier also. Okay. So from the stroma, it will pump the proton to the lumen and electron passes to the cytochrome B6 F complex. From cytochrome B6, electron passes to the plastocyanin and then to the PS1. So this is plastoquinone. This one is plastoquinone. You can write. This is plastoquinone and this one is plastocyanin. Okay. Now, we can understand here how the proton concentration inside the stroma, inside the thylakoid lumen is more than the proton concentration in the stroma. That is how proton gradient develops. Here, the first one, the OEC complex in presence of light rays, it will split the water molecule into proton and electron and oxygen. This electron is transferred to the PS2 for the compensation of the lost electron, and this proton is present in the lumen. Okay. Further, this proton is pumped from the stroma to the lumen. So it also causes increase in the concentration of proton inside the lumen of thylakoid. And here you can see the NADP reductase utilizes proton from the stroma and reduces NADP into NADPH2, which causes decrease in the proton concentration in stroma. So in this way you can see the proton gradient develops across the membrane. That means here inside the lumen, high electrochemical potential gradient, whereas outside the lumen, that is in the stroma, the electrochemical potential gradient is low. Okay. Now this potential gradient is this hydrogen potential gradient is very important. Why? When during the breakdown of this proton gradient, because you know that when the proton gradient is more in the lumen, it must be transferred to the stroma, because the name is chemiosmosis. Chemiosmosis, that means here the proton will be passes from the region of higher concentration to the region of lower concentration. But here it needs facilitated diffusion. Okay, got it? For this membrane having another complex, another system, that is called CF0 F1 particle. This CF0 F1 particle, also called ATP synthase complex. So this one having two parts. Number one is called CF naught part. This is also called transmembrane channel because this part is embedded in the membrane. Okay. So it causes facilitated diffusion. Okay. Whereas another part is CF1 part, which is protruded towards the stroma. So this part having enzyme ATP synthase. That means this one is a catalytic unit, catalytic part. So when the proton is pumped through this channel from lumen to the stroma, while breaking this proton gradient, it will release energy, and this energy will be packed, will be converted into the or stored in the form of ATP with the help of ATPase enzyme or ATP synthase enzyme. So here ADP in presence of inorganic phosphate, it will convert it into ATP. So in this way, at the end of this chemiosmotic osmosis hypothesis, we can see that two things are formed. Number one is ATP and another one is NADPH2. So these two are the product of light reaction, or you may call them as assimilatory power, because these two are utilized during biosynthetic phase or the dark reaction of photosynthesis.